Hello students, welcome to Shore of Sis classes. So here in this lecture we will be starting with the IIT JAM Biotechnology Biochemistry part, and in this lecture we will be mainly focusing on the enzymes portions. So first let me tell you what is the function of enzyme, or what is this term defined, or what we can say when we heard the term enzyme. Enzyme is a biocatalyst. The main term that we can use for enzyme is the biocatalyst. This biocatalyst which are synthesized by the living organism or the living cells. And they increases the velocity or the rate of chemical reaction but these all biochemical functions or biochemical changes occurs without undergoing any change of this enzyme or the biocatalyst. So the enzyme is unchanged after this overall process. Changed after the overall process. So this is the actual definition of enzyme. Now these enzyme can be divided into two parts. So enzyme that can be divided into two parts on the basis of the activity, or on the basis of the how it acts. First is endoenzyme, and the second one is exoenzyme. From the term of endo and exo, we can definitely say that this type of enzyme that acts in the within the cell. It acts within the cell. And the exoenzyme that acts out of the cells, outside of the cell. So this exoenzyme we can also call it as extracellular enzyme. This extracellular enzyme that act outside of the cell. Enzyme. Okay. Now the endoenzyme, normally the plant enzyme, they are all in this category of endoenzyme. Plant enzyme. Now the exoenzyme, mainly they are found in bacteria, fungi and some insectivores like Dorsera, this type of species. So we can classify this enzyme into two parts. One is endoenzyme, another one is exoenzyme. Now the enzyme can be divided into two major classes, and one is simple enzyme, another one is conjugated or complex enzyme. So enzyme can be subdivided into simple enzyme, and another one is conjugated or complex enzyme. Now this simple and conjugated complex enzyme, they are basically subdivided on the base of the amino acid residues, and for the simple enzyme, which are made up of only amino acid, not the other chemical residue. Amino acid is the only portion that the simple enzymes are having in them. But in case of conjugated or complex enzyme which are having amino acid in them, amino acid residue, but along with the amino acid there are some additional non-protein chemical part, non-protein chemical part, and these component acts as the catalytic, this non-protein part having the potential to act as catalyst. Okay, they used as a catalytic residue. So catalytic activity is the function of this non-protein part. This non-protein group that can be inorganic, just like ferrium, just like manganese, like magnesium, zinc, etc. So these are the normal non-protein inorganic factors that helps to act as a catalyst and helps in catalytic activity. And these cofactors, these are the residues that we can call as cofactor. But if there is a complex that is organic or metalloorganic, then that is called coenzyme. Complex that is organic or metalloorganic, that is called coenzyme. So the non-protein part that are inorganic in nature, they are called as cofactors, and when these are organic and or metalloorganic complex, the non-protein part in the enzyme, that is called coenzyme, that helps to the catalytic activity of a enzymatic reaction. Now some enzymes requires both coenzyme and one or more cofactor. So the enzymes cofactor, so the enzyme that requires coenzyme and cofactor both for their activity, and these type of enzymatic reaction, when they are associated with the enzyme part, and this enzyme proteinaceous part is called as prosthetic group. So this is called prosthetic group. Now coenzymes act as transient carrier. This coenzyme act as transient carrier of specific functional groups, just like acyl group, methyl group, hydrogen, hydride, etc. groups. So these coenzyme mainly act as transient carrier. Now a complete catalytic activity enzymes together with the bound of coenzyme or the cofactor is called holoenzyme. Coenzyme plus cofactor with the, so coenzyme and or the cofactor along with the protein part or the active, I rather say the active enzyme, that is called apoenzyme, active enzyme, that is called the apoenzyme, is called together holoenzyme. So this is the simple structure and their simple classification of the enzymes based on their activity, based on their amino acid residue and the protein part, non-protein part, and also previously we have subdivided into endoenzyme and exoenzyme on the basis of how they are acting, where they are acting. Okay, so now So now coming to the nomenclature and the classification of enzyme. Various systems have evolved to name and classify the enzyme. So ultimately there is too much confusion to name a enzyme and to classify on the basis of their structure or their function. So International Union of Biochemistry IUB in 1961 created a systematic basis of enzyme nomenclature and classification. So on the basis of that, there are six major classes, and each with 4 to 13 subclasses, and this is basically based on the system of the reaction catalyzed by the enzyme. The type of reaction catalyzed by the enzyme. So here these six classes are: first is oxidoreductase. Now what is oxidoreductase? These type of enzymatic reaction that catalyzes the oxidation reduction reaction. That is the transfer of electrons between the two substrate. Transfer of the electrons between two substrate, that is called oxidoreductase reaction. Now the major subclasses of these class, the oxidoreductase enzymatic reaction, that is dehydrogenase. The first one is dehydrogenase, that helps into dehydration of the reaction, that removes the hydrogen part. Second one is oxidase, that helps in oxidation. And the third one of these oxidoreductase part is the peroxidase, that added a peroxide group, peroxidase. Then the hydroxylase, option D is hydroxylase. Then the oxygenase, and reductase, F is reductase, that helps in reduction of a reaction. And remember one thing, that when we are giving a name to a enzyme, the second subclass is transferase. Transferase is the enzyme catalyst that transfer the group between the pair of substrate. Transfer the group. It transfer the electrons actually. Electrons transportation is done by the enzyme transferase. Now the major and these class includes the enzyme that transfer aldehydes, ketone or ketonic residues, acyl, glycosyl, alkyl, etc. The third subclass is hydrolases. That catalyzes the lysis of the substrate. Hydrolase, this is the enzyme that add water residue or water molecules, means the hydrolysis is done. Okay, the transfer of the functional group to the water, that is the enzymatic activity done by the enzyme hydrolases. The substrate include ester, glycosyl, ether peptide, etc. Now the fourth subclass of the enzyme is lyase. This enzyme catalyzes the reaction which groups are removed from the substrate. So lyase is the enzyme that removes group from the substrate. Okay. And they are formed or group are added to the double bond. So functional group that are added to the double bond. Now the fifth one is isomerases. Isomerases is the catalytic enzyme or catalytic activity that catalyzes the intramolecular rearrangement. And this intramolecular rearrangement is the rearrangement of atoms and groups and yield isomeric form. So that is why we call this type of enzymatic activity as isomerases, that forms the isomer. The last one is ligases. The sixth one is ligases. Ligases is also called as synthetases. This is the enzyme that catalyzes the formation of the bond between two compounds. So by the enzyme ligase, that is actually the ligation or the it's like a glue. So it's adding two compounds. Okay, that is done by the enzyme ligases. So this is all about the nomenclature and the classification of enzyme. So thank you for watching this video.